Cells can also regulate genes at the level of transcriptional control. This is mostly done through transcription factors. This is probably the best studied area of gene regulation. And the reason for this is just because it's physically easy to study things that happen to be bound to the DNA, particularly things that are at the promoter of genes. We can use prediction tools to kind of guesstimate what would be the promoter of a gene, and we can look to see what would physically be bound at those locations and study them further. So when we're talking about transcription factors, we're talking about proteins. They're DNA binding proteins, and they're going to regulate this gene expression. And so depending on what particular combination that you have, that is believed to regulate activities of different genes. So these are always usually present in the cell, but many of them have to be activated before they're going to bind to DNA. This is going to allow the cell to then respond to different conditions and respond appropriately by turning on other genes, either turning them on or turning them off. So many of these are going to be right next to wherever that gene is. We'll call these the cis-acting sequences. So if something is cis-acting, it's located on the same chromosome, usually pretty close to that particular gene that it regulates. Sometimes it's a bit upstream. If something is acting from a different chromosome or a different location, we'll call that a trans-acting sequence. But most of these will be cis-acting sequences. These will be things like promoters that we just mentioned, as well as other regions such as enhancers and silencers. So there's different types of promoters. Promoters, again, are just nucleotide sequences. They'll serve as recognition sites for this transcription machinery. So in this case here, this would be an example of a focus promoter. There's one particular promoter, there's one gene, and you're going to get one major transcript from this product. You can also have what's known as a dispersed promoter, and this actually may happen more than we realize. It's just a little bit easier to study these types of promoters. But in a dispersed promoter, there is a particular region that is known as a promoter, but transcription may start from any of the start methionines downstream from that particular region. And so you might get multiple transcripts. They may all have different functions, and they may start at this methionine under these conditions, and it might start under this methionine on different conditions. And so this is just something that is still really being studied and really being worked out for many different proteins. When we're talking about these promoters, there's usually certain core elements that they have, and they were going to have an INR or initiator sequence. This is going to start right at the start of that particular transcript. Upstream, we're going to have a TATA box. This is usually -30 to -24 upstream, +1 being wherever that start methionine codon would be. There's a TF2B recognition element, or BRE element, and there could be one, there could be several. There could be downstream promoter elements. This can actually happen within a gene, a motif 10 element, MTE, and so on. And these can all be located either upstream of that particular gene or within the gene itself, usually pretty close to the start. So if you're to look at the gene itself, and you can look to see at the relative transcription level if you were to mutate, say, some of the regions of this particular gene. So in this case, here's the initiation site. Here's the TATA box. Here's some upstream regions. We call them proximal promoter elements, the CAAT, the GC boxes. If you were to do a mutation and you were to block these, you can see the relative transcription level from these particular regions would be turned off quite a bit. The enhancers, again, will be cis-acting, meaning they will be proximal location, so fairly low location, close location. They contain several short DNA sequences. These will increase or enhance the transcription rates. You can be located upstream, downstream, even a little bit of distance from the gene, even within the gene itself. And these are all going to help to start transcription. Silencers are going to be elements that will repress transcription initiation. So in this particular region, maybe something is binding, maybe it will be blocking RNA polymerase from coming and binding to the DNA. Instead of recruiting it, it will physically block it from binding. An example of a transcription factor that I have worked on would be the glucocorticoid receptor. So take a step back. Cortisol is a stress hormone. Hopefully it's not too high levels in students right now. I know towards the end of the semester it can start to rise, especially chronic levels of this. In small doses, cortisol is good for you. In chronic long-term doses, though, not so much. What happens is that this hormone will bind the receptor, the glucocorticoid receptor, that is. That binding of the hormone is a signal for that receptor to go to the nucleus and do something. In this case, it dimerizes, meaning there's two copies of the glucocorticoid receptor that come together, goes to the nucleus, and it interacts with DNA. In the absence of hormone, there is no signal for the receptor to do much, and so it doesn't go to the nucleus, it doesn't bind DNA. And it is usually going to be found either on the cell surface or within the cytoplasm rather than in the nucleus. After giving a cell a hormone, then you're much more likely to find it within the nucleus. Another example is the human metallothionein 2A gene. And there are many different regulatory regions for this particular gene. And so just depending on which of the transcription factors are activated and which of the transcription factors are binding the DNA will change whether or not that gene is going to be transcriptionally activated, whether it's going to be repressed. And this allows the cell then to be able to respond to changes, and some of these can be quite subtle. Changes in the extracellular conditions, changes in the intracellular conditions, and allow the cells to respond.